we want to cover. So let us see. So let us close unrelated window. So this is not related. So cytoscape window here. So basically students they can analyze the data by themselves and they can try it at their home computer and so. So not very big thing. So let us try to understand the what is blast and how this blast analysis is being done. So the PPT, we'll close this one. So we have NCBI PPT, but instead of showing PPT, I think I will prefer to show the practical aspect. What is NCBI site? So firstly, we will observe the site, and then we will try to we will try. We have try this thing. So let us try. Let us see NCBI in short. So what is NCBI and how this analysis? So this is a National Center for Biotechnology Information, and this is website of this one. You can see here all resources, chemical bioassay, data and software, DNA and RNA, domains and substructure, genes and expression patterns, then genetic medicines, genomes and maps, homology, then literature, proteins, sequence analysis, taxonomy, training and treatment variation. So out of this, we want to select nucleotide. So this is our nucleotide. So let's select gal 80 and we want to select east. Let's see if it is coming. This nucleotide, whether this nucleotide is coming or not. So gene name and organism name we are putting. And we are interested in this one, BLAST basic local alignment search tool. So if we have particular gene sequence, can we get information about that gene sequence? Can we know from where that gene sequence has came? Okay, so that information we want to analyze it. So if you put NCBI Google and if you put NCBI in the Google, NCBI. So here you can find the National Center for Biotechnological Information, and this is the BLAST basic local search local alignment search tool. So it will align the sequence and it will try to show the alignment search result. So we have this sequence economics paradoxes GAL80 partial mRNA. So let us see FASTA files. So this FASTA, gene bank and graphic files are the different file formats for this one. Still loading. So you can see on this site. So this is a basic local alignment search tool. So the BLAST find the region of similarity between the biological sequence. So we have one particular DNA sequence, ATGC sequence. So when we say DNA, it is made up of purines and pyrimidines. Purines means adenine and guanine, and pyrimidines means thymine, cytosine, uracil. So if it is a RNA, the uracil will present. If it is a DNA, thymine will present instead of uracil. So this basic local alignment search tool, it finds the similarity between the biological sequences The program compares the nucleotide or protein sequences to the sequence database and calculate the statistical significance. So all things statistically different statistical methods is are being analyzed. So statistical significance. So different way blast can be nucleotide blast. Nucleotide blast means you want to search nucleotide against nucleotide. You put nucleotide sequence in the blast and you want to know the identity of that sequence, that is a nucleotide blast. Then blast X, that is a translated nucleotide. If you put, it will give the protein sequence. So blast X means translated nucleotide sequence you take and it will give protein. T blast N means protein sequence. If you put, it will give the translated nucleotide sequence. Protein blast means if you put the protein amino acid sequence, it will give the proteins, relevant proteins. Okay, so these are the standalone and API blast. So you can download the blast. You can analyze this get blast database and executables. You can do the command line analysis. You can use the blast API. Call the means if you are developing some application, you can use this API. Then use the blast in the cloud for a cloud provider. So specialized searches is there. So there is a smart blast. It finds the protein highly similar to your query. That is a smart blast. Primer blast means you are designing some primer and design the primer specific to your PCR template. For that one, designing primer, you use the primer blast. Global align, compare the two sequences across the entire span. So if you use the Needleman-Wunsch algorithm, you can use the global align. The CD search, that is conserved domain. CD means conserved domain. So particular amino acid sequence is there. If it is conserved, so you can use CD search. IG blast. So this is a immunoglobulin and T cell receptor sequences. So you can search immunoglobulins and T receptor sequences. IG blast, then Wake blast, search the sequences for vector contamination. So if you are doing some molecular biology research, so vector contamination you can search via with the Wake screen. C dot means you can find the sequences with a similar conserved domain architecture. Conserved domain architecture sequence, CD means conserved domain architecture sequences you can search here. Multiple align means align the sequences using domain and protein constraint. So you can do the domain and protein constraints search with multiple align. Mole blast established the taxonomy for uncultured and environmental sequences. So if you want to do the taxonomy analysis for uncultured or environmental sample, in that case you can do the mole blast. So these are the different types of blast you can undertake. So this is a basic local alignment search tool. Okay, so this is a web page of this one, and we want to do the nucleotide blast. So let us click the nucleotide blast. So I have clicked the nucleotide blast, and this is our sequence. So for example, this sequence, or for example, somebody has sent you this sequence in the text file. So this is a text file of this sequence. So this is a text file. Okay, so it doesn't have any name. For example, the instead of Saccharomyces parris is this one. Let us unknown. So this is an unknown sequence, and let us give this file name also unknown. And we want to know its identity. Now it is just ATGC sequence. You can see here it is just ATGC, ATGGACTA. So this is unknown sequence. We want to see from which organism it has come, what is genes, all this information. So if you want to do that, we have nucleotide blast search. So network is somewhat maybe somewhat low, so it is taking more time. Sometimes if many people are using the site, then it is somewhat slow. So you can see here blastine suite. Blastine program searches the nucleotide database using the nucleotide query. So we want the information about this nucleotide, right? So this is our unknown nucleotide sequence. We want to get information about this nucleotide sequence. What we do, we can put this sequence, we can copy this sequence. This is a FASTA file format. FASTA means this is a greater than symbol and name, some name. So this is unknown, so we kept it as such. So we copy it and we paste our sequence as a FASTA. We can enter the accession number, GI number. Accession number means this number you can enter, XM 0. But for example, we don't have accession number, we only have this sequence. So we can enter this sequence here like this. So we have entered our sequence. We have to blast it. Let us blast it. So while blasting, so this is not loading properly still. Uh, Let us blast with default. So gene code. So which gene code we want to apply? Yeast, mitochondrial, echinoderm. So organism you can select, and you can select the other information. So we have sent it for blast. So it has opened in the other page. Let us see if it is loading till then. Okay, this is loading properly. So this is proper look of this NCBI Blast page. So here we have to enter our this one gene sequence we have entered unknown. So this is in the fast format. Uh, this is a nucleotide collection. We want to select organism. We don't want to select organism any standard database non-redundant. We have to select. So here blast. Under the algorithm parameter, we can manipulate maximum target sequence. How many sequences you want? 100 or 5000. This we can give. Short query coverage, expected threshold we can give. Word size we can give. Then match and mismatch you can give. Gap cost we can give. Means if the match is not proper, how should be the match parameter? So these all things we can give here. So complexity, low complexity region, species specific region. So all this information we can give in this nucleotide blast. And if you blast it, now if you blast it, it should show uh, uh, this blast for this one. So let us see if it is coming. So let's see blast result images if it is there. Okay, so these are the blast images. So currently blast is taking quite time, or we can try to minimize the target only 10, and then we'll try to redo it again. Okay, so these are the different things we will get with ENC by blast. So NCBI Blast means, so if this enzyme expected value we can get, it should be quite low. Then identity out of 2633, 2633 is matching, means identity all is identical. There is no gaps. So this information we get. Strand. So whenever we say talk about DNA, it is either a positive or negative strand. So plus plus region we are getting here. So this information we get here, and these are the, this is how result look like. So this is a one organism lactose homo sapiens. So this is maximum score 4000, query coverage 100% same, identity 99% same. So likewise blast we can do, and we can analyze. Currently it is net is not maybe slow, so it is not picking up properly. Let's try to search by query. So nucleotide sequences there. So whenever we say nucleotide, it is purines and pyrimidines, right? Adenine, guanine is the purines, and thymine and cytosines are the pyrimidines. And these nucleotides different arranged with the different sequences in the organism, which we call as a genetic material of the organism, and these things we are analyzing here. So that is a nucleotide blast search. So like these sequences identity we expect, right? So currently somewhat slow. Let us see if it is coming or not. Only a short region we will try to blast. Let us see. This is a Saccharomyces paradoxis. So let us try to keep organism name also. Somewhat slow. So let me explain you result only. Yes, blast result images once more because this search is not internet is quite slow for NCBI site maybe is down somewhat. So I have pressed the blast. So let's see whether it is coming. So till then we'll try to see this one. So this already we are saying expected value should be less. Identity should be maximum 100% identity here. Gaps means here is the CAG. So A should not be missing. If it is missing, the gap penalty will be given. So one gap out of 2633 it will be shown like this. So this is a strand plus the strand. Then this is a different sets of results. Set of result. So there are different blasts. So ideally the result will be like this. So this is a RID job title. This is the nucleotide ID mismatch DNA mismatch repair protein, and this is a DNA mismatch repair protein it should identify. If you are searching DNA mismatch repair protein, it should identify this protein, the maximum score, query coverage, the percent identity, and nucleotide accession number it should identify. So this is a typical result of the NCBI site. So like this result should come. Okay, so currently this site is not working. So there and based on this molecular intensity, you can see different uh, identity. Okay, uh, so these are the things about the blast. Okay, so that's it for today's lecture. So for tomorrow, the guest faculty will be there in the lecture, and the lecture will start as per schedule tomorrow also.